Hey guys, we're here with BIO on the red carpet, interviewing some of your favorite and most popular celebrities.
Today we're going to be asking the popular figures if they know anything about population genetics.
Are your favorite celebrities in the know about bio, or are they simply not?
First up, you know her, you love her, Natalie Portman!
It is such an honor to be here with you today, Natalie. You started a renewed movie series, Star Wars. In Star Wars, there are so many species throughout the series. My question for you today is do you know anything about speciation?
I think I know a good bit about speciation. Well, yes, there are numerous species in the Star Wars universe, but none that can happen without speciation, which I think is something George Lucas definitely had to take into consideration. Well, a species is two organisms that can mate and have fertile and viable offspring, which my friend Michelle Williams would know oh so much about because of her movie Species in 1995. Anyways, within a species, there's both prezygotic and postzygotic barriers. Prezygotic barriers occur before fertilization, and while postzygotic occurs after fertilization, which all forms of speciation occurs through gene flow, which is reproduction from one area to another. Within speciation, there is microevolution and macroevolution. Macroevolution is changes in the allele frequencies in the species over time. Wait, that was microevolution. While macroevolution consists of broad patterns of evolutionary change above the species level. I know more about prezygotic than postzygotic barriers, so let's cover that. Prezygotic is before the zygote. A zygote is a fertile egg. Prezygotic barriers are commonly through reproductive isolation, which is when two organisms cannot meet because of how they reproduce is different. The reproductive isolation prevents hybrids such as ligers, Zorses, or even Ewookiees, Ewok and a Wookiee, from being created. But it does happen rarely. There's also temporal isolation, when species breed at different times of the day, different seasons, or even different years. I mean, a Wampa isn't going to mate with a Wookiee. They thrive in very different seasons, so they mate at different times. Behavioral isolation is another prezygotic barrier, which consists of courtship rituals, or behaviors that draw a mate in. These rituals are different for most species. On Tatooine, it might be showing off your favorite land speeder, while on Mandalore, it might be who has the coolest suit.
Wow, you really know your stuff about speciation and Star Wars.
And there's still more. Within prezygotic barriers, there's also mechanical isolation. This is when mating is attempted but cannot be completed. For instance, imagine a Sarlacc trying to mate with a Lothcat. Wouldn't work too well. That's like me and Chris Hemsworth. Not happening. Gametic isolation is the last one, I believe, which is when the sperm of the species is not able to fertilize the eggs of another species, typically because of a difference in the number of chromosomes of sexes. But I believe the most interesting part when learning about everything prezygotic is the good genes hypothesis, which is typically what we experience as humans. When we see or come into contact with someone we deem as having good genes, they are more likely to be reproduced with, and therefore pass on their good genes, such as Angelina Jolie, who has 6 kids, Eddie Murphy, who has 10. You know, maybe there's a reason Chris doesn't have any.
Wow, I didn't know so much went into the process before mating could even occur. I need to study up on my own questions. And you said that's only half the speciations?
Yes, but as much as I'd love to sit here and chat about this all day, I should probably keep things moving. But do you know who would probably know about postzygotic barriers? Michael. I think they are too far behind me.
We will definitely have to keep an eye out for them. Thank you so much, Natalie. You definitely know your stuff about biology.
Yes, thank you guys.
No, not you, Michael Caton. Hello, Michael Michelle.
Hi!
So I was just speaking with Natalie Portman, and she was super helpful and recommended you to answer a few more questions we had over speciation. She covered everything we need to know on prezygotic barriers. Do you know anything about postzygotic barriers?
Postzygotic barriers. Yeah, of course. To put plainly, postzygotic barriers are said to prevent hybrid zygotes from becoming viable fertile adults, meaning that once a hybrid organism has grown, it won't thrive or be able to reproduce, which eventually leads to hybrid breakdown, where each generation becomes less and less viable.
Wow, yeah, can you imagine a liger running around and reproducing? Do you have anything on speciation that you could share with us?
There's always separation speciations, which consist of allopatric speciations and sympatric speciations. Allopatric speciations is when two groups of geographically isolated, and sympatric speciations is when two groups live in the same environment but have environmental pressures like volcano or sexual selection. Either way, in 10 days, that guy is going to be gone. But for plants, sympatric speciations is called polyploidy, which ends in extra chromosomes. Polyploidy is deadly for animals. Autopolyploidy, though, have more than 2 sets of chromosomes.
You mentioned sexual selection. Can you cover that a little bit more?
Yeah, of course. Sexual selection is when an organism has a preferred mate, looking for certain characteristics, like Matthew McConaughey and blondes, which that selection leads to separate groups and reducing gene pool.
Wow, you know your stuff. Well, thank you so much. I think we've all learned a little something. Well, I think that about covers it all here at the red carpet.
Wait!
Emma Stone?
Yes! She forgot to cover hybrid zones and punctuated equilibria.
Come again?
Hybrid zones and punctuated equilibria. A hybrid zone is an area between species where hybrids can exist biologically. There's stability and there's fusion, like when a dolphin whale lives with other just dolphins and has access to the parent's gene pool. Punctuated equilibria is a very different concept. It happens when there's a sudden change in speciation. Something happens really drastically. Basically just as fast as I fell from the clock tower in The Amazing Spider-Man.
Oh, well, thank you for add-on.
Anytime! Well, now that the red carpet event is finishing up, let's head back to the VIO studio where we interviewed Jackie Mandel. Hey Jackie, it's so nice to have you here in the studio. So the red carpet is coming up in a few days, and before the event, we were wondering if you knew anything on genetics, specifically Mendelian genetics, considering you share a pretty close last name with Gregor Mendel, the founder of Mendelian genetics.
Yeah, actually, because of our last names being basically the same, I definitely have a good amount of knowledge on it. Well, first off, you need to know that a dipod is a living thing that has 23 chromosomes in the sperm and the egg, and that mitosis creates sex cells and gametes when one cell becomes 4. So let's start with true breeding. True breeding is when the exact same alleles cross with each other, leading the cross to give off the exact same genotype and phenotype as its parents. A phenotype is the physical characteristics we can see. A genotype is its genetic makeup. Mendelian genetics also consists of homozygous and heterozygous alleles. The prefix homo means same, so homozygous is when 2 alleles that are in a pair are the same. There are also both dominant and recessive alleles. Dominant are represented by a capital letter, and recessive is represented by a lowercase letter. For example, capital P and lowercase p. Dominant alleles prevail over recessive alleles in phenotypes. Both the dominant and recessive alleles code for a different form of the same gene. For example, a dominant phenotype is brown eyes, and the recessive is blue eyes. Brown eyes will prevail if a dominant allele is present. Both homozygous dominant and heterozygous hold a dominant allele. So, under our last example, if brown eyes was seen, you would know that one of the parents had to have had either homozygous dominant alleles or a heterozygous alleles. But if blue was seen, you know that the genotype or phenotype are recessive. Within Mendelian genetics, and more specifically in phenotypes, there are phenotypic ratios. In order to find phenotypic ratios, we use what's called Punnett squares, which were created by
Chenault Punnett?
Good job. They're used to find genotypic and phenotypic ratios of an offspring when 2 parent alleles are crossed. I think an example would be easier to look at. Let's say that one parent is homozygous dominant and one parent is homozygous recessive. Using a Punnett square, we can learn the chances are the offspring's allele. So, As you can see, our two homozygous alleles have created 4 heterozygous alleles, which gives our genotypic ratio to be 0:4:0, because this would be homozygous dominant, heterozygous, and then homozygous recessive. And then the phenotypic ratio is 4:0 because there are 4 dominant presenting alleles over here and no recessive presenting.
That's so cool. We can almost tell what our future offspring will be.
I wish it was that simple. It would be really easy if it was just dominant and recessive, but there's also complete dominance, incomplete dominance, and codominance. Complete dominance is what we just touched on, when only the dominant allele will show. But it's important to know that dominance does not mean frequency, meaning just because the dominant allele has a chance of showing more doesn't mean it will. Sometimes a gene is not completely dominant or recessive. This is how we get incomplete dominance, when 2 phenotypes show up blended. For example, if a red and white phenotype cross, you may end up with a pink offspring. This is also codominance, when both phenotypes show. For example, a white flower with red dots could occur. Some traits are always going to be more favorable than others. This is where we can have a chi-square test to help us, also known as the goodness of fit test, that you would use of a question basis with very specific numbers and degrees of freedom table, which I do not have on hand. But definitely study up on chi-square Mendelian genetics are so much more complicated than they look on first glance.
That's for sure. But you're really good at explaining things. You really are a great teacher.
It's not my career for nothing.
Well, thank you, Jackie, for teaching us a little something on the Mendelian genetics.
Of course.
Also, in the VIO studio, we interviewed Charlotte Riley, Tom Hardy's wife, and lead in The Edge of Tomorrow, starring Tom Cruise. Hi, Charlotte.
Hello.
So, we've been going around asking a bunch of celebrities if they know anything about population genetics. And our question for you is if you know anything about Hardy-Weinberg equilibrium, since your husband is Tom Hardy.
That's not a question you hear every day, but I do a lot of science fiction movies and shows, so I do know about Hardy-Weinberg equilibrium. It's pretty complicated, for sure. What you need to know is that there are conditions to be in Hardy-Weinberg equilibrium. There can be no mutation, no natural selection, no genetic drift, so no Fonda effect, which is when a population settles into a new area, so there's only a limited number of genes in its area. Or bottleneck effect, which is when a population is whipped out, only leaving a small number of genes to pick from. And there must be a large population in gene flow, either immigration coming in or immigration going out. Now, just to put this out there, no population will ever be Hardy-Weinberg equilibrium, as there is no way to control the environment. Let's go over what everything means in the equation first. P stands for frequency in the dominant alleles, Q stands for frequency in the recessive alleles, P squared stands for frequency of the dominant genotypes, and Q squared stands for the frequency of the recessive genotypes. And lastly, 2P squared stands for the frequency of the heterozygous alleles. There are 2 equations in the Hardy-Weinberg. P squared plus 2PQ plus P squared is equal to 1. And P squared, P plus Q is equal to 1. So, in all, that's how you solve for the Hardy-Weinberg equilibrium.
I have an example right here if you want to see.
This is the full equation, P squared plus 2PQ plus Q squared is equal to 1. Don't mind that, that's messed up. But this is an example where 1,000 individuals out of those 1,000, 360 show a recessive phenotype. So those 360, put that into percentage, and then you'll get 0.36%. And then that's going to be your Q squared, which is again your recessive And so you're going to square root that and get 0.6, which is your p. And remember that p plus q is equal to 1. So to get your p, you're going to get 1 minus 0.6, and then you're going to get 0.4, which is your p. You want to square that to get your 0.16. And then this, you're going to multiply again, this part. You're going to go and plug in the equation, and then you're going to get 0.48, and that's going to be your difference in the area over time.
You weren't kidding about complicated.
It gets easier over time when you do it.
Well, thank you for explaining that.
Anytime.
Well, that sums up this episode of BIO.
Make sure to stay tuned for some more videos so you stay in the know on bio.
Hopefully now you know everything about population genetics.
See you next time.
Bye.